Hello everyone, we are solving biotechnology paper of IIT JAM 2024 and here we are solving the MCQs. The very first MCQ says which one of the following is a simple tissue system in plants. Now the options given are epidermis, parenchyma, phloem and xylem. Your correct answer would be parenchyma. In plant biology, what happens? Tissue systems are broadly categorized into two types. First is the simple tissue system, okay, simple tissue system, and second is the complex tissue system. In the first one, that is in the simple tissue system, it consists of one type of cell which is performing a specific function. In the other one, that is in the complex tissue system, it is composed of multiple types of cells working together for diverse functions. See, I have already chosen the correct answer, but we need to discuss why only this is the correct answer and not the other ones. See, epidermis, let's talk about the very first option, epidermis. While it might seem like a single tissue layer at first glance, but it actually contains several cell types. Like we can say guard cells, then epidermal cells, and it collaborates with the other tissues, like with the underlying parenchyma, for functions like gas exchange and protection. So therefore, it can be categorized as a complex tissue. Okay. Third option is phloem. Oh, before that we need to discuss about parenchyma. Now, parenchyma is that primary plant tissue which comprises of a thin-walled living cells with large vacuoles and extensive intercellular spaces. These parenchyma cells, they exhibit high metabolic activity and they perform various functions like photosynthesis, storage, secretion, and wound healing also. Now, since it consists of only one type of cell, parenchyma is indeed a simple tissue. Okay, it's a simple tissue system. Let's move over to option C, that's phloem. Phloem is a complex tissue along with xylem, and it forms the vascular system in the plants. It comprises of several cell types like the sieve tube elements, the companion cells, the parenchyma, and they work together to transport the dissolved organic materials like sugars and amino acids throughout the plant. Now it's multicellular, okay? Phloem is multicellular in nature and it performs diverse functions and hence it can be classified under complex tissue. Last is xylem. Xylem also is quite similar to phloem. Xylem is also a complex tissue and it is involved in conducting water and inorganic nutrients upwards from the roots. So that's its work, and it also houses various cell types like tracheids, basal elements, then xylem parenchyma with distinct roles in water transport and support. So from this question, we can say that you can remember like this, that the simple tissue systems, it just involve only one cell type with a specialized function, while the complex tissue systems, they combine the different cell types to achieve more elaborate functionalities. Let's move over to the next question. Here is the second question. In DNA replication, the Okazaki fragments are joined by what? In DNA replication, the Okazaki fragments are joined by DNA ligase. Now, DNA ligase, it plays a crucial role in sealing the gaps between the short, discontinuous Okazaki fragments on the lagging strand during DNA replication. So, here we are going to discuss about that only in details. Firstly, the first option, that is DNA helicase, it unwinds the double helix. Okay, DNA helicase, what does it do? It separates the two strands of the DNA molecule at the replication fork, creating a Y-shaped structure. Okay, Y-shaped structure. Second one is DNA primase. Let's take up this one. DNA primase, it lays down short RNA primers. DNA primase, it synthesizes the short RNA primers on the lagging strand to initiate the DNA synthesis by the DNA polymerase. Next is DNA polymerase that we are going to discuss. Now, DNA polymerase elongates the fragments. DNA polymerase adds the complementary nucleotides to the RNA primers on both the leading and lagging strands, thus extending the DNA chains. Okay. Then happens the lagging strand synthesis, which is discontinuous. Now, on the lagging strand, DNA polymerase can only synthesize the DNA in short fragments called the Okazaki fragments. That is what we were discussing till now. Due to the directionality of the DNA synthesis. Next is DNA ligase, which seals the gaps. Now, DNA ligase, our correct answer, correct option, it seals the gaps. DNA ligase covalently joins the 3-hydroxyl end of one Okazaki fragment to the 5-phosphate end of the next, resulting in a continuous DNA strand. So over here in this question, the points that we need to remember is DNA helicase, that's the first option, unwinds the DNA, but it doesn't join the fragments. Secondly, DNA primase, if I come to this, it initiates the DNA synthesis, but it doesn't join the fragments. Coming to DNA polymerase, it extends the fragments, but it can't join the gaps between them on the lagging strand. Last is DNA ligase, our correct answer, which is specifically responsible for sealing those gaps, ensuring that the integrity of the newly replicated DNA molecule. Our next question is the most abundant type of RNA in a metabolically active mammalian cell is which one? It would be rRNA, that is option B. Now, let's start discussing about the other options also. Now, starting with mRNA, that's we call the messenger RNA. Messenger RNA. Now, while it is crucial for the protein synthesis, mRNA, it's present in much smaller quantities than rRNA, okay, due to its transient nature, and it is synthesized from DNA, okay. And it is also used for protein synthesis, and then quickly it's degraded. Next is snRNA. The third the third option given, that's the small nucleolar RNA. A small nucleolar RNA, though involved in processing certain RNA molecules, snRNA are present in very low amounts compared to rRNA. Okay. Next is tRNA. Now, tRNA means what? Transfer RNA. Okay. Now, it is essential for protein synthesis, and tRNA molecules are still less abundant than rRNA. Their role involves carrying specific amino acids to the ribosome for protein assembly. Our last option that needs to be discussed is rRNA. That's our correct option. That is what we call the ribosomal RNA. Ribosomal RNA, it's that type of an RNA which forms the main structural component of the ribosomes. Organelles are responsible for protein synthesis. So due to their constant activity in protein production, ribosomes and their RNA compounds, they are highly abundant in metabolically active cells. So therefore, finally we can say that rRNA, it takes the crown for the most abundant RNA type in these settings. Moving over to the next question. Which organelle in a eukaryotic cell is the site of electron transport chain? Your correct answer would be mitochondrion. Now, electron transport chain, it's a series of protein complexes located within the inner membrane of the mitochondria. And these complexes, they transfer the electrons, releasing energy that is used to create the ATP, that is the adenosine triphosphate. That's the cell's main energy currency. Okay. Now, starting with endoplasmic reticulum or the Golgi apparatus, they are involved in protein synthesis and modification respectively. Okay, now this one is involved in protein synthesis and this one is involved in modification. Coming to peroxisome. Okay. Now, peroxisome are involved in breaking down various molecules such as fatty acids and hydrogen peroxide. So, therefore, the only option that is left with us that correctly identifies the location of the electron transport chain is mitochondrion. Okay. Let's move over to the next question. RNA is a polymer of what? RNA is a polymer of ribonucleotides, nucleotides. Next is glycosides. Glycosides, these are a broader class of molecules that include the nucleotides, but also have other structures with different components. Okay. Next is ribonucleosides. Now, these are the individual units that make up a ribonucleotide, but they lack the crucial phosphate group which is needed for linking the nucleotides together in a polymer. Next is ribosis. In ribosis, what happens? These are the sugar molecules that form part of the nucleotide. So, but they are not the complex units which are needed for polymer formation. So, therefore, while glycosides, ribonucleosides, and ribosis, they are all important components of the RNA, but the correct answer is ribonucleotides, as I have chosen over here, because they possess the complete structure which is required for linking together and forming the RNA polymer. Okay, so we are going to keep it till here in this video. Let's meet again in the other videos. All the details about our classes are given in the description box below.